Good day, everyone. I am Arjun Umbas. I am for today's reporter. Today, I will discuss to you the five topics, but first, I will discuss the origin of life. So we all know that origin is life is a topic that has intrigued scientists, philosophers, and thinkers for centuries, which is all about understanding how the first forms of life came into existence on Earth. So, abiogenesis is a theory that explains how life on Earth originated from inanimate matter. When we say inanimate matter, it is a thing that is not alive or object that has no life. Abiogenesis is a complex process that likely took hundreds of millions of years. Abiogenesis also refers to the theory and the process that lead to the appearance of typically chemical compound that lacks of carbon-hydrogen bonds that is not an organic compound. Example of inorganic substances are ammonia, hydrogen, sulfide, metals, and most elements such as calcium. Abiogenesis theory also suggests that life originated on Earth itself with the right conditions and chemical reactions, leading to the creation of simple organic compounds which eventually formed early life forms. So in conclusion, the most accepted ju- so in conclusion, the abiogenesis is the most accepted theory, but it's still a theory. There's a lot we don't know about the origin of life. Scientists also continue to study about this subject. So the Miller-Urey experiment shows and presents that the planet was covered with a vast primordial soup, which was a mixture of methane, ammonia, and the hydrogen. Energy from the sun, lightning, and Earth's own heat triggered chemical reactions that created more complex organic compounds. So before I will show you the example of Miller-Urey experiment, let us let us first know the scientist behind this experiment. So Stanley Lloyd Miller, born on March 7, 1930, he is an American chemist who made important experiments concerning the origin of life, demonstrate wide range of vital organic compounds that can synthesized by fairly simple chemical processes from inorganic substances. Harold Clayton Urey, born on April 29, 1893, he is an American physical chemist whose pioneering work on isotopes. He earned Nobel Prize in Chemistry in 1934 for discovering of deuterium. He also played a significant role in the development of the atom bomb as well as contributing to theories on the development of organic life from non-living matters. Harold Clayton Urey was the professor of Stanley Lloyd Miller. Miller are one of his students. So the Miller-Urey experiment is under abiogenesis since these scientists were able to produce fundamental organic compound for living beings from inorganic substances under laboratory conditions. So this visual shows the example or present the Miller-Urey experiment. I have a video to show to you to further understand the experiment of how the first life forms. So as I've said earlier, this experiment is under abiogenesis theory.
Miller designed an apparatus to simulate the ancient water cycle. Together, they put in water to model the ancient ocean. It was gently boiled to mimic evaporation. Along with water vapor, for gases of the atmosphere, they chose methane, hydrogen, and ammonia. These are simple gases which scientists at the time thought were probably abundant on the ancient Earth. They added a condenser to cool the atmosphere, allowing water molecules to form drops and fall back into their ocean like rain. The ancient Earth would have had many sources of energy: sunlight, geothermal heat, and even thunderstorms. So they added sparks to the atmosphere to simulate lightning. Experiment for just 1 week, their ocean became brownish black. Careful analysis revealed that through a series of reactions, many complex molecules had been produced. Among these were amino acids, special molecules of life that we once thought could only be built inside the bodies of living creatures. This was a pivotal breakthrough in science, so significant in fact, a rich collection of complex molecules produced by natural chemical reactions. In this soup, further reactions could take place, eventually producing living cells. At the
So, from this image, we can see that from the simple organic compound and amino acid polymerize into enzyme, and then after it forms a complete living cell, which is cell is the basic structural and functional units of all living organisms. Here's another video to furthermore understand the experiment and see how the existence of cells forms.
In this apparatus, they placed water, ammonia, methane, and hydrogen, all of which was exposed to heat as well as an electric current meant to simulate lightning. After about a week, they found that the reaction mixture contained a number of different amino acids, which, as we remember from biochemistry, are the building blocks of proteins. Since proteins include enzymes, which catalyze the formation of nucleic acids like RNA and DNA, this offered good evidence that it is indeed possible that the basic materials needed for life could have been generated spontaneously. Furthermore, this experiment was only a week long, so 500 million years of molecules sloshing around in the primordial soup could conceivably produce much more impressive results. These findings prompted further speculation as to the specifics of how the first primitive cell could have formed. Understanding of biochemistry, it's not much of a stretch to assume that the basic organic components delivered from the Miller-Urey experiment were able to polymerize and were then serendipitously encapsulated in a lipid bilayer, which we know can form spontaneously because of hydrophilic interactions between the polar heads and water molecules and hydrophobic interactions where the non-polar tails hide away. And there you have the first protocell. This first cell must have been incredibly primitive compared to even the simplest ones today, and subsequent cells grew in complexity over time. This was due to the ability of nucleic acids to self-replicate, as well as mutate, which is what led to the evolution of all the diverse kinds of life on Earth over the eons.
So, speaking of cells, what is cell? So cells are the basic structural and functional units of all living organisms. They are often called the building blocks of life because all living things from the smallest bacteria to the largest animals are composed of one or more cells. Cells is term cell was coined by Robert Hooke. Observe a honeycomb-like structure under the microscopic while cutting a thin section of cork. Hence, he named the structure as a cell. So, there are parts of a cell. It is the cell membrane. It is a thin, flexible barrier that surrounds the cells, separating its internal environment from the external environment. It regulates the passage of substances in and out of the cell. Then we have cytoplasm, a gel-like substance inside the cell membrane. where various organelles are suspended. It contains water, salts, and organic molecules. Next is we have nucleus. It is in eukaryotic cells, a membrane-bound organelle that contains the cell's DNA. The nucleus controls cellular activities and contains the genetic information necessary for cell growth, development, and reproduction. So, endoplasmic reticulum, or ER, is a membrane-bound organelle found in eukaryotic cells, including plant cells, animal cells, fungi, and protists. It is a network of interconnected membrane that extends from the nuclear envelope throughout the cytoplasm. There are two main types of endoplasmic reticulum: the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So, a rough endoplasmic reticulum has ribosomes attached to its surface, giving it a rough appearance under a microscope. The rough endoplasmic reticulum plays a key role in the synthesis, folding, and modification of proteins. Second is the smooth endoplasmic reticulum. It lacks ribosome in its surface, giving it a smooth appearance. It involves in lipid metabolism, including the synthesis of lipids such as phospholipids and cholesterol, and the metabolism of carbohydrates. It also plays a role in detoxification processes within the cell, including the metabolism of drugs and toxins. So the endoplasmic reticulum, it is a dynamic organelle involved in various essential cellular processes, including protein synthesis, protein folding, lipid metabolism, and detoxification. Its structure and function can vary depending on the specific needs of the cell and the organism. So we have also Golgi apparatus or Golgi complex. It is another membrane-bound organelle found in eukaryotic cells. It consists of a series of flattened membrane sacs called cisternae. The Golgi apparatus receives, modifies, sorts, and ships proteins and lipids synthesized in the endoplasmic reticulum. The proteins and lipids enter the Golgi apparatus in vesicles derived from the ER within the Golgi. These molecules undergo further processing, such as glycosylation, additional of sugar molecules, and sorting into vesicles for transport to various cellular destinations. Golgi apparatus or Golgi complex involved in the secretion of proteins, the formation of lysosomes by packaging enzymes, and the synthesis of cell membrane components. Next part of cell is membrane receptor. Membrane receptor are proteins located on the cell membrane or within the cell that bind to specific signaling molecules such as hormones, neurotransmitters, or growth factors. So, receptors play a crucial role in cell signaling and communication, allowing cell to respond to external signal and regulate various cellular processes. Next is mitochondria, are double membrane-bound organelles found in eukaryotic cells, often referred to as the powerhouse of the cell. They are responsible for generating adenosine triphosphate, the primary energy currency of the cell, through the process of cellular respiration. Mitochondria have their own DNA and ribosomes, allowing them to partially self-replicate and synthesize some of their proteins. We have also the nucleus. Nucleus. So nucleus, as what I've said earlier, nucleus is a membrane-bound organelle found in eukaryotic cells, which are cells with a through nucleus enclosed within a membrane. It is often referred to as the control center of the cell because it houses the cell's genetic materials and controls cellular activities. So, function of the nucleus are storage and replication of genetic materials. Nucleus contains the cell's genetic material in the form of DNA. So DNA carries the instruction for building and maintaining the cell and is organized to structures called chromosomes. During cell divisions, the DNA is replicated to ensure that each daughter cell receives a complete set of genetic information. Also, second function of nucleus is the gene expression and regulation. The nucleus is involved in the process of gene expression, whereby the information encoded in DNA is used to produce an RNA and proteins. These processes includes transcription, the synthesis of RNA from DNA, and translation, the synthesis of proteins from RNA. So gene expression is tightly regulated to ensure that the right genes are turned on or off at the appropriate times and in the correct cell types. Also, assembly of ribosomes. Within the nucleus, the nucleolus is responsible for the synthesis and assembly of ribosomal RNA and ribosomal proteins into ribosomes, which are cellular structures involved in protein synthesis. So, in overall, the nucleus plays a critical role in controlling cellular activities, maintaining genetic integrity, and transmitting genetic information to subsequent generation of cells. So, there are three components that compose the DNA. First is the phosphate group. Second is the deoxyribose sugar. It is the second component of DNA. It has a 5-carbon sugar that forms the central part of the nucleotide. It is attached to the phosphate group at one end and to a nitrogenous base at the other end. So the deoxyribose sugar provides the structure for the nucleotide and help from the backbone of the DNA strand. Nitrogenous base, this is the third component of DNA. Also, there are 4 types of nitrogenous bases found in DNA: the adenine, guanine, cytosine, and thymine. These bases are attached to the deoxyribose sugar and project inward from the DNA backbone. The sequence of nitrogenous bases along the DNA strands forms the genetic code which carries the structure for building and functioning of an organism. So the phosphate group is a polymer made up of nucleotides, and each nucleotides consists of 3 components. One of these is, as what I've said, the phosphate group, which is a molecule containing 1 phosphorus atom bonded to 4 oxygen atoms. The phosphate group forms the backbones of the DNA molecules, linking the individual nucleotides together through phosphodiester bonds. So, the 3 components of DNA together form nucleotides, which are the building blocks of the DNA molecule. So, based on the image that will show here, adenine pairs only to thymine and guanine to cytosine. So there are three types of RNA. So RNA is a versatile molecule involved in various cellular processes, including protein synthesis, gene regulation, and the transmission of genetic information. There are several types of RNA molecules found in cells. First is, as we can see, the messenger RNA. Messenger RNA carries the genetic information from DNA to the ribosomes, where it serves as the template for protein synthesis. Molecules are mRNA or messenger RNA molecules are transcribed from specific regions of DNA called genes during a process called transcription. So after transcription, mRNA undergoes processing including the addition of a 5-capped and a poly A tail, as well as the removal of introns, A non-coding regions, to form mature RNA, mRNA. So the mature RNA molecule contains codons, which are sequences of 3 nucleotides that encode specific amino acids. These codons are read by ribosomes during translation, where they specify the order in which amino acids are assembled into proteins. Second is we have ribosomal RNA or rRNA. It is a structural component of ribosomes, which are cellular organelles responsible for protein synthesis. Along with these proteins, form the two subunits of ribosomes, the large subunit and the small subunit. Ribosomes use rRNA or ribosomal RNA to catalyze the assembly of amino acids into proteins based on the sequence of mRNA. So rRNA molecule helps position from mRNA and the transfer RNA molecules, which carry amino acids during translation. Lastly is the transfer RNA or tRNA. These molecules play a key role in protein synthesis by bringing amino acids to the ribosomes during translation. So each transfer RNA or tRNA molecule carries a specific amino acids at one end and has an anticodon at the other end. The anticodon is a sequence of 3 nucleotides that is complementary to a codon on the messenger RNA. So during translation, transfer RNA molecules recognize specific codons on the messenger RNA through base pairing between their anticodons and the codons of the mRNA. So, transfer RNA molecule is bound to the appropriate codon on the messenger RNA. Its attached amino acid is added to the growing polypeptide chain, forming a protein. So these three types of RNA work together in the process of proteins synthesis, which mRNA carrying the genetic information from DNA to ribosomes, RNA providing the structural framework for protein synthesis, and tRNA or transfer RNA delivering the amino acids to the ribosomes for incorporation into the growing polypeptide chain. So lastly, I will show you a video.
Messenger RNA starts off in a cell's nucleus. Here it transcribes or copies the instructions for assembling a protein from a portion of the cell's DNA. Next, the messenger RNA takes these instructions or protein recipe
to a ribosome in the cytoplasm. The ribosome, which is made out of ribosomal RNA and proteins, is the site where this recipe is read and translated into a protein. Finally, transfer RNA in the cytoplasm brings specific amino acids to the ribosome so that the particular protein encoded by the recipe can be made. We'll cover more detail about the way DNA and RNA make protein.
So that is all. I hope you understand how the origin of life or the first life exists. Thank you for listening.